So thank you so much for joining us today. I'm thrilled to be joined by Dr. Anastasia Khvorova and Dr. Vignesh Hariharan and my colleague, Tim Freeman. Just a couple of housekeeping things. So this is a Zoom webinar. It's not a Zoom call, given the number of people that are on. So you can see and hear us, but we can't see or hear you. There will be an opportunity to ask questions, and you should be able to see the Q&A at the bottom of your screen. So we will handle questions at the end of the presentation, but I'll be reading your questions and we'll answer as many as time allows. Also, RSRT is hosting this webinar together with Cure MECP2 Duplication Syndrome, which is the next phase of the 401 project. Okay, so Dr. Khvorova is a world-class scientist at the University of Massachusetts Medical School. Her expertise is in oligonucleotide, I've said that a million times, oligonucleotide technology and therapeutics. And she's part of the world-renowned RNA Therapeutics Institute at University of Massachusetts. Her postdoc, Vignesh Hariharan, is a molecular biologist, and he is the lead on this MECP2 duplication project. And Tim is our chief development officer. He has a daughter with Rett syndrome. We do have some exciting news to share, which we'll do at the end of the scientific presentation. So do please make sure to hang on till the end. We're envisioning about a 30-minute presentation followed by Q&A, and then we'll make this announcement. Okay, Dr. Khvorova, if you would like to share your screen.
I would love to, if you allow me to do that.
Let me allow you.
You have to click on my name.
Here we go. Here we go. Yep. You know, Zoom keeps adding more and more layers of protection. There you go. You should be able to do that now.
Yay, fantastic.
Okay.
Can you see my screen?
We can.
Okay, thank you so much, Monica and team, for inviting us to talk about a very exciting project in the lab. And the project is related to developing next generation of oligonucleotide therapeutics for modulation of MECP2 expression and MECP2 duplication syndrome. And as Monica mentioned, my lab is located at fourth floor of this beautiful new building, and we're around 20-25 people, which is a mixture of organic chemists, oligonucleotide chemists, molecular biologists, cell biologists, and pharmacologists, advancing this breakthrough technology towards clinical administration. So when we start to talk to Tim Riley and eventually Monica about whether this can be applied for MECP2 duplication syndrome, the challenge is there. And what you see over here is kind of a cartoon representation of the healthy child, which has one normal X chromosome with one MECP2 region. And then if this particular region is mutated, then what you can see is that half of the neurons stop expressing MECP2, and this results in half of an amount of MECP2 protein in the brain, and this causes Rett syndrome. And if you have a duplication of this region, which is MECP2 duplication syndrome, you have too much MECP2 protein, which resulting in a very similar clinical manifestation. So the problem is that we need to fine-tune the expression of MECP2 in a duplication syndrome where we silence it approximately 50%, but not more than 50% because we don't want to generate Rett syndrome. And this represents a very interesting, challenging, and technically motivating project. So another kind of justification behind the project that we work on many other neurodegenerative disorders which result in neuronal death. And what is beautiful about MECP2 duplication and Rett syndrome, which offers some hope that while it changes dysregulation of the brain, it doesn't cause neurodegeneration. So neurons are still there. And in mouse models, in adult mice, mouse models of Rett and MECP2 duplication syndrome, the dosage normalization results in reversal of some of the phenotypes. So the vision or conceptual idea behind this project is to develop an RNAi-based therapeutic for disease-modifying treatment of MECP2 duplication syndrome. And if you look at it from a patient perspective, what do we need? We need to have robust delivery throughout the brain because you need to silence it pretty much in every single neuron. You have to have non-burdensome dosing regimen where raising a child like that is super cumbersome, and it has to be something which is realistically feasible. So we're looking for long duration of effect and ideally dosing regimen where the drug needs to be administered every 6 months or every 12 months. The most challenging technical part is we need to have partial but uniform modulation of MECP2. And right now the technology has advanced enough that we can silence it 100%, but achieving partial modulation is very interesting. And of course, we need to have a robust safety profile to expand the treatment immediately post-diagnosis. So what is RNAi? We kind of use this word freely, and this is what is currently revolutionizing the field of human medicine. It's how we think and treat diseases in the future is going to be different. So RNA interference is a natural mechanism, like every single cell has this mechanism expressed, but what can happen, it can hijacked by artificial small double-stranded RNAs and reprogrammed to silence disease-causing genes. And in this case, we're trying to hijack this fundamental biological mechanism to target MECP2 and reduce its expression and normalize its expression to a normal level. So it's a fundamental biological mechanism, and as a result of it, it can potentially induce a very long-lasting effects with a single drug administration, and it seems to be safe. So as any technology, and people who work in IT fields, they have seen this curve a lot. RNAi-based drugs went through this hype where their mechanism was discovered. UMass professor, RTI Therapeutic Institute professor Craig Miller got the Nobel Prize for discovering of this mechanism. All big pharmas jump into it. Things are being moved into clinic and nothing works. Like everything fails in clinic. There's all the money gets out of the field. There is a disillusionment that nothing is working, but chemists like myself and many others continue to advance this platform forward, which has resulted in first drug approved in 2018 and unprecedented pharmacological properties of this type of drugs. So at this point, the delivery problem is solved and those drugs are approved for one organ only, which liver, where a very complex chemical scaffolds, which are using multivalent GalNAc conjugates. I don't want to go into details, but it's a scaffold optimized for liver delivery. They're causing 6 to 12 months 6 to 12 months duration of effect. I think Lisa has raised hand. How do you?
No, we'll do questions.
Okay. Okay,
Yeah.
perfect.
Look,
So 6 to 12 months duration of effect, imagine it, yeah? And what was most exciting is that there is effectively no reportable adverse events with this drug called inclisiran, with exception of injection site reaction. And it's expected to get approval end of this year. And because of the value, transformative value of this technology platform, and in this case, this particular drug is normalizing levels of cholesterol in the blood, which probably every single person over the age of 50 should be taken from now on. It was bought by Novartis for $9.7 billion. So it worked, and it works amazingly well. So it gives you this exaggeration duration of effect with a fantastic safety profile. But it works only in one condition: when you can deliver it robustly and sustainably to the tissue of interest. So oligonucleotide therapeutics and siRNAs belong to this new class of informational drugs, meaning that as long as you optimize a chemical scaffold, which gives you robust delivery to a particular tissue, you can reformat it or reprogram it, the scaffold, to target other genes in the same tissues. And right now for liver, there are around 40 to 50 clinical candidates in development utilizing this inclisiran chemical scaffold, which gives you robust and sustained modulation of gene expression in liver. So what my lab does is optimizing this chemistry and delivery to get delivery to other tissues. And of course, to treat Rett syndrome, Huntington, Alzheimer's, MECP2 Duplication Syndrome, the key is developing chemistries which gives us ability to deliver it to the brain. So oligonucleotides do not cross blood-brain barriers. They just do not. So, but what we can do, we can administer them directly to CSF with intrathecal injection and see whether we can achieve some distribution in the brain. So my lab has been working in this field for a very, very long time. And what you see over here in blue is a slice of the mice brain, and blue is just, it's depicting every individual cells. And then we inject oligos and we label them in red. So whenever you see a red color, it means that the drug is present in the brain. And we screen a lot of different conjugates trying to optimize the CNS delivery platform. As you can see is that we can, if we increase height of obesity, we get great retention, but very limited distribution. So this wouldn't work for Rett because we need, or MECP2 Duplication Syndrome, because we need to have it throughout the brain. Or if we reduce height of obesity, we get slightly better distribution, but little retention. So we were screening a lot of, and I think it's kind of a very interesting story because it tells you about the role of serendipity in scientific research. When we normally present or write papers, we think we present things that we're geniuses, which have thought about things and we plan it this way and we achieve something. In reality, serendipity is very often is a driving force behind the transformative scientific discovery. So the story is kind of funny because what we were doing, we were making those siRNAs and small RNA molecules, and we are touching different moieties to see whether this will give us a good brain distribution. And one of the moiety we were thinking about is vitamin D, and I can give you a long lecture why. And there is a postdoc in the lab, very good chemist. His name is Matthew Hassler, and right now he's head of chemistry at Atlanta. And he was challenged to synthesize this compound. And he was doing it for quite a while, and eventually we have animals ready. We are injecting a lot of different variants, and I'm asking him, Matt, do you have the vitamin D conjugate? And he's kind of saying, you know, I kind of do, but it's not really clean because it's very hard to perform this reaction. And we said, okay, fine, it's not toxic. Let's just inject and look at the distribution. So when we injected this, what is supposedly was vitamin D, we were jumping up and down, because what you can see here, this is a brain, and then green is neurons, and you can see this red color, which is depicting the drug in every single neuron. So we are excited enormously because we developed this novel chemistry, which gives us distribution. But there is many different compounds. So each peak represents a different chemical compounds. And it took us another year to figure out what was giving us the good distribution. And interestingly, it appears that it's not the vitamin D. It's not the unconjugated compounds. It's when accidentally, because of bypassing reaction, two oligos, two siRNAs were conjugated together, increasing their size and promoting cooperativity and interaction. This is where we got this outstanding, unprecedented distribution throughout the brain. So we call this divalent scaffold as CNS RNAi, and when you inject it in CSF, you get robust distribution in the brain throughout every single region of the brain. And you can, when you inject it in CSF, you can look at the silencing in the different brain regions. And in this case, we were targeting gene Huntington, which is causing Huntington disease. And the only thing I would like you to see here is that there is supposed to be bands here, yeah? If we treat with a control, there are bands corresponding to Huntington protein. And when we treat to the drug, it's gone, yeah? You cannot detect it. And this duration of effect, like this elimination of the Huntington, is lasted for extremely long periods of time. Even 6 months post-injection, we're still seeing modulation of Huntington expression. So what I would like to mention here is that in a context of a MSP2, we don't want that, yeah? If we do this, we'll get a Rett syndrome. We want 50% modulation, which represent an additional challenge. So the next thing, okay, we can silence it in a mouse brain, and this was exciting. But if you compare the size of the human brain and mouse brain, it's quite different. And so what we need to do, we need to ensure that we get uniform distribution, not only through the tiny brain, but also to the large brain. And to do that, we use a different model of disease, which is called sheep and monkey. And indeed, when we take those divalent scaffolds and we injected it in a sheep, we get a pink sheep brains. And at that point, we felt that we are up to something, yeah? And you can see over here, this is a control brain. The brain is universally pink, yeah? So you get oligos throughout the brain and the spinal cord. And in clinic, this type of things can be delivered by intrathecally or intraventrically. And we compare two routes of administration, and essentially they look the same. So finally, we got money donated to us to run a couple of non-human primates, a very limited amount of animals. We injected them with a drug. We see universally pink brain. It's distributed throughout the brain. I'm just showing you the section of the cortex where you can see that every single neuron, like uniformly gets this red color indicating that the oligo is delivered. And when we see it both neurons and astrocytes, we see it accumulation throughout the brain. And most importantly, again, what you're looking here, it's elimination of this band, which in this case is Huntington. And you can appreciate that we silence it in the cortex, we silence it in the caudate, we essentially silence it throughout the brain. So that is exciting, and this compound was out-licensed to a new company at Atlanta, funded by Fidelity Ventures, and they're moving it to clinic formally. But another very interesting parameter of the technology was that at 25 milligram in non-human primates, we have seen no toxicity. We look at cell counts, we look at the biomarkers talking about activation. We did an MRI, which is for academic lab it's a super expensive way to look at the tox, but it really gives us an idea whether we see any edema or any inflammation, and we have seen practically none. So the last thing we did, we look at the specificity, and again, it's a very complex experimental design. But what you look over here is a graph, which each dot represents another gene and genome. And if you go right, it's upregulated. If you go down, it's downregulated. And if you go at a Y scale, it gives you a significant. And you can probably appreciate that in this particular case, there's one gene which is really downregulated and modulated, which is Huntington. Okay, so at this point, we were absolutely, the article describing this new technology platform was published in Nature Biotechnology. We made it to the cover of Nature Biotechnology, which is one of the most prestigious biomedical journals, because we are opening CNS for intervention. So we developed this chemical scaffold, which gives us robust and safe way to modulate gene expression in CNS. And as I mentioned to you, the siRNA is an informational drug. So we did it for Huntington, we did it for APOE, we did it for C9orf72. And at this time, Vignesh joined the lab, and he was looking for the things to do. And I was asking him, what do you want to work on? What is your life choices? And he said, I would like to make a drug and cure disease. And, you know, for a postdoctoral fellow joining the lab, it's pretty ambitious goal, but we are living in very interesting times. And I said, okay, fine, let's try it. And at that point, we had a collaboration with Monica and others trying to work on Rett syndrome. And they approached me and said, Anastasia, but can you silence MECP2? And I'm saying, you know, silencing is much, much easier than in reactivation in half of the neurons. So Vignesh joins the lab, and his goal is get it done. And the problem is, a technical issue is we have to silence it, but we don't want 100%. We need just fine-tune the expression. So how would you do it? And Vigi came up with two possible strategies how to do it. So I already have shown this slide where we're taking our drug and we load it in a RIS complex, and then it's finding a target and cleave it. And Vigi's ideas was to figure out how would you chemically modify this drug substance so it still can load into RIS complex, but it doesn't do this reaction very efficiently. So the cleavage rates are reduced. And I think he was able to do it successfully. And another idea Vigi came up with is that if you look at the literature, MECP2 is actually expressed as two different isoforms. So there's two variants of this gene, which slightly differ in their sequence, really, really slightly. But if you take a Rett syndrome model, so the model where there is no MECP2 expression, and you just express one of those variants, this is sufficient to recover the disease. So Vigi's ideas was, let's just silence one of those isoforms and see whether this would be sufficient to give you partial modulation of MECP2 expression. And because the compound is selective only towards one isoforms, we are not being in a risk of silencing it too much in some regions of the brain and causing Rett syndrome. So two strategies: advanced siRNA chemistry to support partial 50% reduction, and then fully silence one of the isoforms. So the first thing Vigi did, he did the screen. We designed a lot of compounds using advanced bioinformatic means. He did it in a screen in this chemical scaffold, which is expected to have load into RIS complex efficiently, but do not cleave it fully. And he identified multiple leads, and some of them, luckily for us, is targeting both human and mouse. And this is important from a standpoint of development and validation. Maybe it's not exactly 50%, maybe it's a little bit more, but we are getting where we want to be. And 2 compounds were identified in particular, which gives us a very nice and potent but incomplete modulation of MSP2 expression. So the next thing what Viggy did, he synthesized it as this divalent scaffold, injected them intraventrically, and look at the level of modulation of MSP2 expression in a mouse brain. And what he looked at multiple different regions. And as you can see, he's getting there, yeah? This is our controls. The lower the bars is the lower the level of the expression. And with a blue compound, he's getting around 50 to 60% modulation on average. So it's maybe a little bit too much. And with a red compound, he's getting 45% modulation, maybe a little bit not enough, but we're getting within a range. And what was very exciting about this data is that with this new advanced chemical scaffold, he's indeed getting uniform modulation of expression, but not a complete modulation of expression, which is exactly what we need to achieve for treating of MSP2. And in this case, for those 2 compounds, it's either 55 or around 40%. And at this point, we probably would like to move a blue compound forward, but we have a portfolio of different compounds with which fine-tuning MSP2 expression to different levels. So another thing which Maggie did, he asked a question as, would it sustain, this partial modulation will sustain in a case of exaggerated delivery? Like in a case of the liver, if we synthesize it as a gallneck, we theoretically should be able to silence at 99%. And indeed, when he injected it in a pretty high dose, yeah, very high dose, it's 20 mg per kg, he silenced MSP2 in the liver, but he only silenced it partially, indicating that we are indeed achieving this plateau of downregulation. So the last thing which Viggy actually executed on his idea of partial silencing, he developed a screening system which allows you to look at the different isoforms of MSP2. And what you see over here, it's a screening for identify compounds which will silence E1 isoform, but not E2. And as you can, E2 is in gray and E1 is in red and black. And you can see there is a panel of compounds which is just silencing E1, but not silencing E2. And when he has done those responses, he now have several candidates which are very selectively silencing one isoform, but not another. So this is a complementary strategy. In this case, we're looking at a different chemical scaffolds, the one which will silence the gene completely, but we are only targeting one isoform, but not 2, to ensure that we are under no circumstances will silence the MSP2 completely in certain neurons, potentially causing Rett syndrome phenotype. Okay, so this is where we stand today. Viggy has optimized lead siRNA compounds showing partial 45-55 modulation of MSP2 expression in mouse brain. He also has optimized lead siRNA compounds which are selectively silenced in just one MSP2 isoforms. This is validated in vitro only. We're doing in vivo studies as we speak. So what is next? The next is we need to demonstrate and compare those lead compounds in a MSP2 in a model of MSP2 duplication syndrome in mice. We have to demonstrate that we are partially silencing, and this is reducing the phenotypes and allowing neurons to recover. We have to demonstrate efficacy and safety in non-human primates. And practically, we would like to see that even in large brain, we can achieve this uniform but partial modulation of gene expression. And then the next step we'll be doing formal preclinical development, which is, GMP manufacturing, IND enabling talks, and designing the clinic and moving those compounds forward. I also have to say that we formed a company around a year ago where this novel chemistry platform, divalent siRNA, have been out-licensed. It's co-founded by Craig Melua, who is a Nobel Prize winner, and it's funded by Fidelity Ventures. It's in stealth mode, but its lead compound clinical candidates are in Huntington disease. So with that, I just want to say that it takes a village to get something like that done. And I'm really honored to work with a talented group of chemists, cell biologists, pharmacologists, oligonucleotide chemists. We are funded by many different institutions because what we do is expensive. And in particular, this project was funded by Rett Syndrome Research Trust, and Viggy did all the work, which is all the work, ideas, execution, design, which is related to applying this transformative technology platform towards development of disease-modifying therapeutics for MECP2 duplication. And I think that's pretty much it. And I'm happy to answer any questions you might have.
Great. We do have a couple of questions, but I wanted to just make a couple of points first. First of all, that was a great presentation. And it's recorded, so we'll get it online at some point. So if people want to hear it again, most of the audience, they're not scientists. So, you know, it helps to hear it a couple of times and digest it. But I wanted to make a couple of points. So the animal work that was presented are normal mice. And so I just want to make the point again that the next step is to repeat the experiments, but to do them in the MECP2 Duplication Syndrome animal models. And that would be done at Jackson Lab, which is a nonprofit in Maine that is kind of the gold standard repository where everyone deposits their mouse models after they've published them, but they also conduct research themselves. And it would be done under very, tight operating procedures and very rigorously done, just as if it was a clinical trial in people, right? So that needs to happen. And then you mentioned, Dr. Khvorova, that non-human primate experiments would follow the mouse model work. And I just want to make it clear that that's done in normal non-human primates, not a disease model. And that's typical in drug development.
And I would like just to mention that with non-human primates, it's kind of a complicated question because we're silencing it 50%. And this is not exactly what's happening with Rett syndrome, because with Rett syndrome, you have half of the neurons which have no MECP2 expression and half of the neurons which has a normal expression, approximately. So it was interesting to see is that in a preliminary Vignesh study in a wild-type mice, and I understand it's only 1 month, but Rett syndrome, it's such a severe phenotype that you see it almost right away. So 50% modulation of MECP2 throughout the brain did not cause any significant, let's say, any robust clinical phenotypes. Of course, we didn't measure the details, but if you silence MECP2 completely, you can see it right away. So it's might be safer than we think, but it's something which need to be experimentally validated.
Good. And the other point I wanted to make is to, unlike gene therapy, for example, gene replacement that you're delivering with a vector and you hope would be a one and done. Of course, that has advantages, right? One and done means you don't have to continue to give the treatment. This would be ongoing treatment, but the advantage of that is that you could potentially fine tune the dose.
I think it's absolutely correct, Monica. And to some degree, I am actually looking at gene therapy as well and oligonucleotide therapeutics as complementary technologies to each other. With the gene therapy, the scary thing is you do it once, there's no coming back.
Right.
And if you have not, if something is screwed up, yeah, if your scientific hypothesis was wrong, you're done. Like there is no reversal. The gene is edited out or it's supplemental. There's no coming back. With oligonucleotide therapeutics, you can run, it's more like a large small molecules, extremely long lasting small molecules, because when we're talking about 6 months or 12 months duration effect, it's not 24 hours. So it will take a while for effects to come back. There are mechanisms how we can develop antidotes, and we actually do it routinely now where we can stop, it's called antagomers. We can stop the function of the drug by delivering of the antidote. So from that perspective, clinically, we can test a lot of hypotheses. And for example, if we see is that Rett MECP2 partial modulation is safe and effective, and if by that time AV delivery platforms, it's advanced in the ways that you actually can do it throughout the brain, and you can do it in regulatable fashion or fine-tunable fashion. Fantastic. You can follow up with a gene therapy drug, and then you are sure that it's going to be safe and you're once and done. And hopefully this is how things will evolve, but it will be many, many years for gene therapy before it's fine. It can be tuned and distributed widely.
Okay, so let's, there's a couple of questions, so I'll read those out. So one question from Christopher. I have to say these are pretty sophisticated questions. Mm-hmm. Does each isoform have the exact same function, or is there a possible negative outcome in completely silencing one of them?
Viggy, how about you answer this question? Because I know you researched it a lot, and Viggy has like 20 slides on this topic, which I all eliminated because Monica told me that we need to keep it short. Yeah.
Right. The short answer is no, that silencing one isoform over the other would not lead to any negative effects, because so far studies have shown that one isoform can do the function of the other. So in a sense, they are both redundant, and even if one's not there, the other one can do the function. More detailed answer, the isoform E2 has a higher DNA binding affinity, which means it can perform functions much better, but in the normal body, it's present at a much lower concentration. So essentially, you have two things that can do the same thing. The highly potent one is at a lower concentration, and the isoform that's less potent is at a much higher concentration. And what we're looking to do is silence the less potent one, which is at a higher concentration, but silence it much lower. And so far, literature tells us that the second isoform, which is more potent, will actually be able to take over. And that's there in the slides that Anastasia showed previously.
So I also have to say, if you look at the literature, and again, we need to test that in a context of, let's say, non-human primate, wild-type non-human primate, and validate that you silence one isoform completely and the animal is fine. You have to do this experiment. It's a must. But in literature, what we see is there is a lot of developmental regulation of two different isoforms. But during embryonic development, but after the birth, they seems to be more or less normalized. So the differences are less. But it's a fantastic question, and we need to experimentally test it.
Okay. The next question is from Samir. He says, "Great talk. I have two questions. In the ICV injection of siRNA into the mice, how old were the mice?" Let's answer that one first.
Viggy.
Or were they adult? He wants to know.
Yes, these are adult 6 to 8 week old mice. They're
Okay.
wild type, which means they are normal.
Okay.
They're full-size mice. They're fully grown, sexually mature.
And then he'd like to know, how does the siRNA specifically knock out the E1 isoform? Where is it targeting in the MECP2 locus?
That's a great question. The way we design our siRNA, and I think Anastasia is pulling up the slides right now, different isoforms actually have a different sequence, at least in some parts. So isoform E2 has an extra region called exon 2 that isoform E1 does not. And this actually contributes to different potency, maybe different effects in different brain regions. But essentially, there's something unique about isoform E1 that is not there in isoform E2. And we're able to selectively target it because siRNA are informational drugs which target a specific sequence. So as long as there's a unique sequence, we can target it and have selective activity or downregulation.
I think when you look at the slide, which shows you the difference between two different isoforms, so practically they utilized a slightly, this piece of information which is different, which is called E1 and E2, it's part of the exon 1 and 2, is different. So here it's red and here is magenta. So when we target just this region, we will silence this isoform, but not the other. So this is a strategy.
Okay, great. And then this is a question from Xavier. How long is it to begin to see improvement of the healthy mice? Apologies for my bad English. Greetings from France. So
we have not done this experiment.
That's what has to come next, Xavier. They haven't tested it yet in the disease model of the mouse.
So what is known, there is a beautiful paper and you can look at it, look at this paper. It's published, sorry, I'm having, it's published, I think in, I think over here, it's published in Nature. It's called Reversal of Phenotypes in MSP2 Duplication Mice Using Genetic Rescue or Antisense Oligonucleotides. Antisense do not deliver everywhere and it's need to be continuously infused to see any efficacy. But with the genetic rescue, it seems to be that within 2 months of doing so, they start to see gradually the phenotypes recovering. I have to say, and we had this long discussion with Monica, and I think it's very, very important to understand it with Monica and other people in MSP2 duplication syndrome, that as with any neurological disorders, the brain is such a complex organ, and clearly it's also very adaptive. So if the brain is developing in a context of the constant damaging signal, like having too much MSP2, it will adapt and develop in a certain way. And whether it's possible to reverse this damage which is done through by being developed or established in a context of this negative signal, by elimination of the negative signal, it's a huge clinical question. And maybe I just want to be conservative to say I think full recovery is very unlikely. But, and probably the earlier you do that, the better is going to be the outcomes. But maybe this more requires comments from team from a development perspective. But this is kind of unknown, and I think expectations is that there might be some recovery, but it's very unlikely that just by elimination of their toxic signal, you would be able to completely reverse years of abnormal brain development. Team, what is your opinion about that?
Well, Tim's our fundraiser, so he
Okay.
probably doesn't have an opinion
Okay.
about that. But I think we get asked, you know, we talk about this in Rett syndrome as well. What would a cure look like? We don't know. We don't know until we have something that we can actually test in a clinical trial. I think common sense tells you that intervening early will always be better. You know, intervening in a 1-year-old would be better than a 30-year-old. I think every parent of an older child kind of, you know.
So I will just quickly say, what is a proposal?
We don't know until we actually do it. And I think, you know, the fact that there's no neurodegeneration in either duplication or Rett, and the fact that we get this robust reversal in mouse models, you know, gives us tremendous optimism.
So what is the proposed delivery method of the drug? At this point, I think we are most likely will go with intrathecal injection, which is outpatient procedure, if maybe every couple of months, and then depending how it goes, it can be more less rare. If it will need to be eventually repetitively administered, most likely you will put some kind of a catheter in there, so it's not invasive.
I think with the rest of the questions, because we need to move on to the next section. I will work with Viggy and you, Anastasia, to provide answers, and we'll email the answers out.
Okay,
so
perfect.
We'll address
Thank you.
Questions. And you're free to get on with your research if you like, or you can stay on, whatever you prefer, but we're going to talk a little bit about how we're going to fund the great next steps that you need to do. So thank you so much for participating in the webinar. Okay, let me share my screen. Tim, I'm going to turn this over to you.
Okay, thanks. Can you hear me okay?
Yes.
Yeah. Hi, everyone. I'm Tim Freeman. I'm RSRT's Chief Development Officer. My role is to help families raise the funds that make this research possible. It cannot happen without funding. So we have an exciting announcement today that Monica alluded to at the very beginning of the call. The Baker family, who live in Maryland, are pictured here. And they are left to right, Ashley and Amy, Dave, and on the far right is Megan, who has MECP2 Duplication Syndrome. So about a week ago, Amy and Dave contacted me and said they wanted to make a $50,000 donation to this research, but they wanted it to be a challenge match that would inspire other duplication families to donate themselves and to fundraise from their networks. So today we're announcing the Baker Family Challenge Match. And I want to thank the Bakers for doing this. It's not only obviously very generous, but it's also motivating, I hope, to all of you. And it's a reflection of their confidence and optimism in this research. So we need to raise $330,000 for this project that you just heard about. And the way the Baker's Challenge works is that they've told us that if the duplication community can raise or give $280,000 towards that $330,000 goal by the end of this year, the Bakers will kick in the final $50,000. So it's a $280,000 goal, really. And that's an ambitious goal to raise by the end of the year, but I really think you can do it. And this is really a terrific opportunity to make the dollars that you raise count for even more. But you've got to act now and fundraise from your networks. So these funds are raised quickly by the end of the year. I can't overemphasize that enough. The next slide.
Sorry.
That you might ask me, well, okay, Tim, how do we do that? How do we take advantage of this terrific match that the Bakers have offered. So this slide shows different ways to fundraise, ways that we have been proven really the most effective ways to raise funds for the research. The first one is crowdfunding on our platform called Rett Give. It couldn't be easier to set up one of these Rett Give campaigns. They're personalized campaigns, and all you need to do to start one is email me and tell me you want to start a campaign, and I'll send you some very easy instructions for some materials to send me that include a couple of photographs and a very short paragraph. And again, the funds that you raise on Rett Give will go right to this duplication research. The second method of fundraising here is Facebook crowdfunding. Also super easy, very effective, especially if you have a big group of friends on Facebook. The web address is here. Be sure to include the word duplication in the title of your campaign so that it goes to the duplication fund. Another method of raising funds is to write a letter or an email to people in your network. And this is especially effective, I think, if you have someone in your network who is philanthropic, who has the capacity to make a significant contribution. Sometimes crowdfunding might not be quite personal enough for someone who is capable of making a very large donation. So a letter, an email, it works. And I'm here and very happy to work with each of you on those letters individually and help you draft it. So call or email me anytime you want. And the last way to raise money, of course, is to donate yourself. So I want to add one more thing, and some people have expressed some concern to me about fundraising in the age of COVID-19. And I will say that, yes, we all need to be thoughtful about our fundraising. And I don't approach people in my network who have been directly affected by COVID. But what I found is that the vast majority of people are sitting at home and are really looking for something to get behind, something to support. I saw a poll recently that said about philanthropy in the US, and it said about 7% of people plan to decrease their philanthropic giving this year. 25% plan to increase it, and the rest, which is about 67%, plan to give the same that they've always given. So it's a tiny percentage of people, a small percentage of people whose giving philanthropy is affected negatively by the current environment. People want to be supportive. They want to give something to something that's promising, that's urgent, but they do have to be asked, and they have to be asked by the duplication community. So I am here to help with any of those methods of fundraising. I love hearing from families anytime. Please call me, email me to get started on any of this. And I think you can do this. It's an ambitious goal. We can do it. It is so important. It's worth spending the energy on to take full advantage of the Baker's terrific match. So thank you.
Great, thanks, Tim. I just see some questions here about that I think I want to address, just because they talk about likelihood of recovery or not. And someone says here, we understand that this therapy will not reverse duplication. I think what Dr. Khvorova was saying is that in order to manage expectations, that we not to expect perhaps a complete recovery, okay? And I'll use my daughter as an example. She has Rett syndrome. She's 24, almost 24. I'm not expecting a cure for her like it never even happened. But I do think it's possible to have dramatic improvements and to have progress for the first time in her life with a therapeutic that could address the underlying root cause of the disease. And I feel the same way about duplication. There's a lot in between, having duplication or having a complete cure. There's a lot of area in between that, and it's not all or nothing. So of course our goal is a cure for everybody, but if we can't achieve that, maybe it'll just be newborns, maybe it'll be under a certain age, but maybe there can be dramatic improvements for everybody else. The bottom line is we don't know until we take these programs and these therapeutic strategies forward. And Dr. Khvorova acknowledged the Rett Syndrome Research Trust for funding, and that's accurate, but in actuality, it's all of you. It's the duplication community that raised funds to develop a mouse model, to do the reversal experiment, and to fund the other type of foundational work that's gotten us to this point that we are at now, and we can't stop now. So I think this is incredibly important work with the potential to be very translational. This is not a basic science experiment. Okay, this is work that could potentially develop into a drug. So it's exciting, and I hope that you'll digest the information and get involved, because the duplication families have to raise this money to get it done. So Tim and I are here to answer questions. We'll get to the questions that we weren't able to answer via email, and as soon as we get the recording, we'll be able to share it with you. So thank you, everybody. Thank you, Dr. Khvorova, Dr. Hariharan, for joining, and thank you so much for your important work. Thanks, Tim. Okay, goodbye, everybody.
Thank you, Monica.